Welcome everyone. Thank you for your time. Thank you for your attention. My name is Fabian Gerlinghaus. I'm the co-founder and CEO of Cellares. At Cellares, we're working on accelerating access to life-saving cell therapies. And I'll be giving a brief overview about the company and our technology and how we're making a contribution to actually solving this problem. So we're very inspired and very excited about all the progress that hundreds of cell therapy companies are making in this field to actually push forward the therapeutic and clinical, ultimately commercial potential of cell therapies for patients. And this slide speaks for itself. We now live in the age of personalized cures. Cell therapies have shown and proven their potential to actually cure very aggressive types of blood cancers, and I'm personally incredibly excited about applying that same potential to solid tumors as well. The problem that we're focused on at Cellares is really scalability amongst other things. Today, cell therapy manufacturers are actually unable to meet patient demand at commercial scale. If you're summing up all of the patient doses produced between all of the FDA-approved drugs, the entire industry produced about 4,000 commercial doses last year, while the addressable patient population, according to McKinsey, is at 450,000 patient doses, and they expect that to exceed 2 million patients within the years to come. So there was also an article in Stat recently, some of you may have seen it, talking about the inability of pharma companies to meet commercial scale patient demand. And that article quoted that 20% of the patients who are eligible for CAR T-cell therapies are actually dying on the wait list because they don't get a manufacturing slot assigned to them in time. So that's tragic, right? We've got drugs on the market that would most likely save their lives, yet people are still dying. Hence the pressing need to resolve the scalability challenge here. The problem is going to get much more pressing over the years to come. At this point, there's about 2,700 cell therapies in development. With all of these drugs coming down the pipe, the FDA projects that they'll be approving 10 to 20 new cell and gene therapies every single year starting 2025. And then adding on top of that, cell therapies are increasingly moving up the treatment plan. And as we're moving up the treatment plan as an industry, that obviously opens up the patient population even more into the tens and ultimately hundreds of thousands of patients per year per drug in some indications. So hence the need for scalable cell therapy manufacturing technologies. The issue from a manufacturing perspective today is really cell therapies, for the most part, are made with benchtop equipment that usually handles one unit operation for one dose at a time, and lots of manual labor in between, on the order of 50 manual operating steps. And all of these manual operating steps, you're very vulnerable to operator error and contamination. Oftentimes, you've got open transfers of the cells and reagents from one consumable made to work with one instrument made by one vendor to the next consumable made by another vendor for another instrument. So try and copy and paste this process in an autologous setting 10,000 times, 50 manual operating steps, 10,000 processes. A lot of opportunities for operator error. Meanwhile, we're generating on the order of 400 to 600 pages of paper-based batch records for every single dose, right? That's a book being written for every single patient. And now do that 10,000 times. That's a lot of documentation overhead. That's a lot of time spent doing that. And in the logistics involved to actually manufacture just, say, 10,000 patient doses are pretty mind-boggling. Very quickly, this becomes unmanageable from a logistical perspective, not to mention the fact that it's very hard to hire all the talent that would be required. So cell therapy companies urgently need manufacturing technologies that are fully automating the process from start to finish in a very high throughput fashion. That's what's required to make this scalable. And they also need manufacturing technologies that make the entire manufacturing process super reliable, very robust, cost efficient, and do all of that without compromising on flexibility. The process is the product. We need to provide the flexibility to support a wide variety of different cell therapy manufacturing workflows. So this is a sneak peek at what our solution to this problem looks like. It's called the Cell Shuttle. And what makes the Cell Shuttle platform scalable is really the combination of end-to-end automation and high throughput. So we've developed a fully closed, fully automated single-use cartridge. I will say this is the most sophisticated single-use cartridge ever built in the life sciences. It's about the size of a carry-on suitcase. And it's so sizable because it actually contains all of the different technologies that are usually made by different vendors for different instruments in one closed tubing set to actually execute the entire workflow from start to finish. So what you load into this cartridge at the beginning is the starting material from the patient or the allogeneic donor. And what you unload from this cartridge at the very end of the process is a cell therapy product that's ready for release testing and infusion into the patient. And then importantly, you can load up to 16 of those cartridges into the Cell Shuttle, and in this way manufacture 16 batches simultaneously per instrument. So that's either 16 different autologous products or 16 allogeneic batches. So that's what makes us scalable. In our opinion, it's going to make a big contribution to actually helping pharmaceutical companies and biotechs address patient populations at commercial scale. A little bit more detail on the cartridge in terms of workflow steps that we're supporting. It really starts with enrichment of white blood cells, red blood cell depletion. We're supporting concentration steps, buffer exchanges, cell selection, activation, both viral and non-viral gene transfer. So we've got both capabilities within this cartridge. Cell expansion, fill finish, and formulation. Typically, we're formulating, in the case of an autologous product, into 4 bags already located on the cartridge, 1 for infusion, 1 for backup, 2 for quality control. And by virtue of integrating all of these different processing steps in one closed tubing set, we're eliminating the risk of contamination. And then this is a cartridge that's made for automation from the get-go. So there's a whole lot of automated interfaces between the cartridge and the corresponding instrumentation. So you don't have any manual setup steps where you would need to manually weave tubes into pinch valves or anything like that. And then in terms of the overall workflow, how do you use this platform in general? Step 1, you design your specific cell therapy manufacturing process in software, specifically in our process design studio, and I'll be elaborating on that in a little bit in more detail. And then once you've designed your workflow, you load the starting material into the cartridge, and you load the cartridge into the Cell Shuttle on the front. And from this point on, it's actually hands-off, fully automated. It's true walk-away automation. So you can come back 3 to 14 days later, depending on the length of the process that you've designed, and at the very end, pick up a cartridge that contains the final drug product. As I mentioned, we can do 16 cartridges, 16 batches simultaneously, and that provides a tremendous amount of throughput. So throughput depends on the length of the process. Something like Kymriah, a 14-day process, you can make about 400 doses per Cell Shuttle per year. And if you have something much shorter, T-charges between the 2 and a 3-day process, that gets you up to about 2,000 doses per Cell Shuttle per year. So at that point, you'd need 2 Cell Shuttles to produce what the entire industry made last year in terms of number of batches. We're eliminating about 90% of the human labor, which is really required given the dearth of cell therapy talent. The risk of operator error, and also all that talent that exists in our industry would be much better leveraged actually developing new therapeutics rather than repeatedly executing the same unit operations manually. And we're about 90% more space efficient compared to the manual methods that the system is replacing. So I got to speed up here a little bit. Big picture benefit here is we're reducing the process failure rate from about 20%, that's ballpark what we're seeing in the industry today, to less than 5% by automating and closing the process, we're on average reducing the cost of goods sold by about 50%. The numbers vary between 30% and 70%, depending on which process specifically are we looking at. But we're seeing very significant cost savings for every process that we've looked at for every partner company. And the big one is scalability, the ability to run 16 batches simultaneously. So if you run the numbers, in order to make 20,000 patient doses, and this is based on a 10-day autologous T-cell-based process, you'd need about 45 cell shuttles. 45 cell shuttles fit into a 110,000 square foot facility. If you attempted to do the same thing with manual methods, you'd need 1.4 million square feet. That's not one facility, that's a dozen facilities. And then CapEx goes down significantly because you don't need to build out 1.4 million square feet anymore of facility space. You need to build out a 110,000 square foot facility. And headcount goes down from about a theoretical 2,600 employees to less than about 400 employees. And really the big one here is that it makes it possible in the first place to produce 20,000 patient doses per year with this platform. Flexibility is the name of the game. Our software platform really allows you to design your own processes, execute those processes, monitor those processes, and you get a reporting solution that actually auto-generates electronic batch records, mostly automatically, because the software knows what it did to the product at what time. So we're transitioning from paper-based to electronic batch records and also automating the generation of that. Double-clicking here on process design. Our process design studio gives you a library of cell therapy manufacturing unit operations that you can quite literally drag and drop into the order that you need them in. Double click on any of those unit operations and you get access to all of the underlying process parameters for all unit operations. So we're really opening it all up, including non-viral gene transfer parameters, so on and so forth. So this is really the first platform that gives you best of both worlds. Automation, which is what you need to actually scale out and meet patient demand, in combination with the flexibility that you need to actually run your specific process, the process that defines your product. And traditionally, the only way to get that level of flexibility was with benchtop instruments handling one unit operation for one patient at a time. And that's why most of the FDA-approved drugs that are on the market are made in this way. But it locks you into a manufacturing paradigm that is not scalable and not cost efficient. And what we're doing here is we're bringing together best of both worlds. So this platform supports about 90% of cell therapy modalities, basically any suspension-based workflow. We're supporting autologous and allogeneic workflows. We're reagent agnostic. You can load any reagent into this platform. And we're agnostic to the whole debate around centralized versus decentralized manufacturing. You can deploy this platform in either fashion. We've automated the act of taking samples. It's a parameter that you specify in software. How much of a sample do you want to draw? When do you want to draw it? And then you can either have that sample picked up on the backside of the instrument for analysis on any third-party instrument of your choice, or you can also have it auto-analyzed on our integrated QC platform that gives you cell count, density, viability, metabolites, nutrients, pH, dissolved oxygen, gases. So all of this data shows up in a dashboard where you've got all the processes that are currently running on the cell shuttle. You double-click on that dashboard and any of those processes there, and you get to see where are we in the process, all the data that we've collected, all the in-process data plotted over time, and 45 seconds left. I'll make this point here. This is a platform that doesn't just provide value in commercial scale manufacturing, but it's also very valuable at the very beginning of your drug development process, because instead of running 16 full-scale patient doses, you can define an arbitrary number of different cell therapy manufacturing workflows with different permutations of your process parameters and run 16 different experiments in parallel on the cell shuttle. And you don't need to do that at full patient scale. You can do that at a fifth of a leukopak, keep your process development and your reagent costs low. But importantly, you're doing it on the very same platform that you're going to use to meet all of your clinical production, your commercial manufacturing needs, same consumable cartridge. Same software, same instrumentation, same technology. That's not what happens today. What happens today is an academic institute comes up with a brilliant cell therapy. They out-license it to a pharma company. The pharma company goes, brilliant, but we need to industrialize this. We need to make this more scalable, more robust, more cost efficient. And then they change a few of the manufacturing equipment. The moment you do that, triggers so much process development work, so much comparability studies, that it keeps departments busy for years. And all of this work goes away. So if you adopt this platform early on, in the preclinical phase, and you do your process development on this platform, that actually accelerates your timeline to FDA approval quite significantly. And I'll leave it there for today. Thank you very much.